This is TWIS. This Week in Science, episode number 782, recorded on Wednesday, July 15th, 2020. Why Friendliness is Fabulous. Hi, everyone. I'm Dr. Kiki. Welcome to This Week in Science. Tonight, we will fill your head with cancer cures, pig blood, and friendliness. But first, thanks to the Burroughs Welcome Fund and our Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer. The information you're hearing from your religious, business, and or political leaders about the deadly viral pandemic may or may not represent the actual evidence-based understanding of scientists and medical researchers who know what the expletive they are talking about. If viruses could be prayed away, the pandemic would be over by now. If political will sticking to your guns against popular opinion could persuade a virus, the pandemic would be over by now. If pool parties getting back into the office and reopened restaurants was intimidating to the virus, the pandemic would be over by now. If we just took science seriously, if we acted a little sooner or waited at home a little bit longer before going out, the pandemic would be over by now. But instead, we may as well await the ringing of a bell and the voice from the street calling out, "Bring out your dead! Bring out your dead!" The only thing that may yet save us from the fate of death by ignorant powers that be is This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, everybody.
Good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again to talk about all the science that happened this last week and maybe a little bit more, because we just like to put that little extra in there because it's science, which is always extra. Okay, on the show tonight, I have stories. I have gas. I have a story about gas.
Oh, I'm sorry.
Yeah, anyway.
Story about
Pig blood. The pig blood. I brought the pig blood, and we are joined by a guest tonight to discuss the benefits of being friendly. We'll be introducing Dr. Brian Hare in just a little bit here. Hello, Brian.
Hi, guys.
Justin, what do you have for the show tonight?
I have cancer cures that work, presidential daddy issues, and unpickling your brain with pickled capers.
Pickled, pick a peck of pickled. Blair, what's in the animal corner?
Oh, I have lefties and righties in fish. I have bystanding rats, and I have a little bit of seahorse news.
I love seahorses. I'm looking forward to that one. So let's do some show here. As we jump into This Week in Science, I would like to remind you that you can subscribe to us on all podcast platforms. Just look for us. Look for This Week in Science, and you should find us there. You can also find us on YouTube and Facebook. You can go directly to our website, TWIS.org, to listen to episodes and see show notes. All right, let's dive into some quick science stories. Finally, cosmological agreement. It's happening. It's here. Data is in from the Atacama Cosmology Telescope, the ACT. It mapped the cosmic microwave background radiation of a large portion of the night sky, and the results from its measurements agree with previous results from the European Space Agency's Planck telescope survey, which determined a specific measurement of the Hubble constant. The Hubble constant is a measure of how fast our universe is expanding, how fast stuff is running away from other stuff in the universe. And this is great news for the validation of cosmic microwave background as a credible measurement method, but it does not solve the discrepancy between measurements that are being made using the cosmic microwave background measurement versus other methods like light from stars and stuff. And so there's this thing called the Hubble tension, which it doesn't solve. It doesn't fix that at all. It's just kind of sets it more starkly as a thing that is tense and could also indicate a problem with the standard model of physics. What, Justin? You're looking cranky-faced.
Well, yeah. So it's just hard to measure things that are really far away. I mean, it's just difficult. I don't think we have to quite get to the point where we have to, this start over with a new model. The standard model is something wrong. It's just hard.
I think this attitude, maybe this gives me some insight into your state of mind right now. Tell me about
the scale of the universe is really big.
It's big.
Yeah.
It's vast and even.
I think it's a long way to the corner store.
I think everyone can agree on that too. It's big.
Yeah.
All right, tell me about some cancer cures that actually work. Is that a
So this is a clinical trial evaluating a sample of one. They tried out in this novel, recently kind of popular immunotherapy treatment for a child with a form of muscle cancer that had spread into the bone marrow. This is getting to that point of no return, right? Showed no detectable cancer following treatment. No detectable cancer. This was in the muscles, this was in the bone marrow, and now the child is cancer-free. Of course, the treatment was the chimeric antigen receptor T cell treatment. This is the CAR T that we've been talking about. These are T cells that are engineered, in this case, to target the HER2 protein on the surface of cancer cells. The trial was conducted by researchers at Baylor College of Medicine and is currently or very recently in Nature Communications journal. So this was a very high risk child of not surviving very much longer when they initiated this trial, which is what made them a candidate for it, because you don't just experiment on children. And this isn't the first time they had tried this. So they, in previous clinical trials, they had found that their treatment was having positive outcomes, but not curing. They were reducing in a sort of subset of patients. And when they looked at it, what they kind of decided the problem was it wasn't persisting. These new engineered T cells were going in, but they weren't persisting long enough to really tackle. They were knocking down the cancer in some, but they weren't quite getting the job done. What they did was kind of interesting. This new wave of it is they used a little bit of chemotherapy to basically wipe out pretty much eliminate the existing immune cells. They knocked down the existing T cells and upped the amount of these engineered T cells that they put in. And basically it worked. And then 6 months later, the cancer returned. They went back and did again. They repeated the trial on the same child. And at this point, it's been 19 months and they are completely cancer-free still. And it's interesting. That's also, so this is a quote from one of the authors, Dr. Nabil Ahmed, who is also associate professor of pediatrics and immunology at Baylor. The study shows that CAR T cells could perhaps be acting as vaccines by exposing cancer proteins to the patient's immune system. With more understanding, further refinement of the design, CAR T cells could be effective against some currently incurable malignancies.
Get in there, get the body to help fight off whatever's growing that shouldn't be there. Get it to recognize it. Yeah.
Yeah, and it started with, this is, whenever people are like, oh, genetic engineering, we're going to use genetic engineering on children in the future. Yeah, the future's today, and it's curing cancer.
Good.
It's a good thing.
Let's keep doing it.
Yeah,
let's make it so let's turn it into a process that isn't only for the most the most deathly ill. Let's turn it into a process that can work for other people so that people don't get to the point where they are that sick. That's where we hope we can be some way.
And that's where the clinical trials. So there were trials before that showed it was going to be, safe. They weren't having some of the CAR T therapies of the past did have some potential really negative effects, which is why they only have been using them on people who are deathly ill. Because in some of the earlier trials, people were dying from the treatment. Small percentage, but still enough that you wouldn't do it for somebody who is otherwise healthy, but down the road is going to get worse. So, but the more that they improve, you might go in, and the treatment happens quick. It's like within 48 to 72 hours, like the reversal takes place. The tumors it's insanely quick.
Like,
so you could,
it's done.
Yeah, you could go in on to the doctors on a Friday with cancer and Monday be cancer free. That's how insane the future is going to be.
It's brilliant. It's not insane. It's brilliant. And it's wonderful. And I want to dream of it because it's a happy, happy dream of the future. And I'm going to hold on to this. Okay. Blair, tell me about seahorses.
Oh my goodness. Well, what's so famous about seahorses? What do you always think of when you think of seahorses?
The two seahorses kissing and then they make a heart.
Besides
their little tails wrapping around seaweed and they hang.
What I think about is that they're, as far as we know, the only vertebrates that have male pregnancy, or them and their relatives, like leafy sea dragons and stuff. So what's really cool about this story is that University of Sydney found that seahorses actually have a really specific connection between them and their babies during the male pregnancy. And this is through something that is basically a placenta. They're actually transporting nutrients via placenta to their babies. It's not exactly like a human placenta. They don't have an umbilical cord. And so they need to do further work to figure out exactly how it's working. But as far as they can tell, the same way that human mothers transport nutrients, oxygen, and immune modulation, they do all that in seahorses as well from the father.
But it's the daddy who has the placenta.
Yeah.
Does it come from the babies like it does in mammals?
We don't know.
An interesting question.
Yeah. So that's the question. Where's it coming from? Like what hormones are going on inside seahorse fathers? Because you would assume that placenta formation, these sorts of things are pretty strongly linked to things that we consider fundamentally female. But clearly, that might not be the case.
So this is my question.
I mean, it's fundamentally the parent who has the babies internally. Fundamentally.
Can I just ask a question? Did somebody just decide which was the male and which was the female seahorse and just got it wrong? And for some reason, nobody's fixing it?
Justin, if I may, one makes sperm and one makes eggs.
Okay, I guess that. I mean, that's one definition, I suppose.
That is one definition.
We just picked that one and have stuck with that. Okay, that's fair enough.
Yeah,
I guess.
We like naming things. We're human. That's just. I will give you a name, and now you are named. Yes, you shall be. Sometimes though, we get gassy, which seems to be the last 2 decades, unfortunately.
I'm so sorry.
driven by
to Marshall and Kai mostly, but
driven by increases in agriculture and in natural gas. Methane emissions have reached 10%. A rate of increase of 10% over the past 2 decades. They have increased, reaching 596 million tons in 2017. This is pretty much due to the increased consumption of red meat globally, especially in Africa, which was interesting to the researchers. And it's very tied to these agricultural emissions related to cattle ranching, except for in Europe, where European countries have been working very hard to reduce red meat consumption and cattle ranching. And also, they've made efforts to reduce landfill emissions. And so Central Europe's emissions have been going down compared to everybody else's, but the rest of everybody is pretty much negating that benefit.
So the thing with the methane, if I'm correct, and I'm probably am not, is methane does not persist in the environment as long as carbon does. Does not persist as long as carbon in the atmosphere. However, has a magnitudely larger effect.
Yes,
like a magnified
correct.
effect than carbon on greenhouse. So we heat up quicker with methane than we would under carbon, but it won't have a 50-year cycle. It'll be much shorter. Exactly.
So if we methane sticks around for about 12 years, and that's a lot shorter than carbon dioxide, but
carbon dioxide is like 50.
Yeah, 1 ton of methane is the equivalent of 85 tons of carbon dioxide. So the fact that our methane emissions right now, during a time when we're trying to consider emission reductions to stop global warming, this is potentially a short-term problem because it also feeds into these feed-forward cycles where you have increases in heating that lead to the release of more methane, that lead to the release of more carbon dioxide, that everything, it gets worse.
Right, because there's a tremendous amount of methane underneath the frozen tundras of Siberia and Canada. And as those melt, those release, we accelerate faster with the heating, more melt. If it was just a methane problem though, it's sort of weird, but we could all take those steps that need to be taken and be done with global warming in 10 years.
Mm-hmm.
The problem with the carbon cycle is it's 50 years. So I don't even know what year it is. 1970 was 50 years ago? Is that right? 1970 is 50 years ago?
No. No. I don't want to think about that.
Okay, sorry. I know that kind of hit home too. This is why I paused. I'm like, wait, that's almost how old. Oh God. But the global warming. The global warming we're largely experiencing now is the carbon that started in 1970.
Yep. If you put it in those terms. Yep.
It's even longer than that, Justin. It can be up to and over 200 years. Most of it stays about 50 years, but it can stay for hundreds of years, carbon dioxide.
Right.
But it's we had the muscle cars thing going on. No, I guess that's more 60s, huh? Yeah, the 70s was when we got the small cars again.
It's more that there's carbon dioxide in our atmosphere right now from the Industrial Revolution. Just think about that. Goodness. Yeah.
Anything that we do right now, we're not going to see results until the end of this century, if not beyond this century. So we better get to work, everybody.
Mm-hmm.
Not just methane. All right. Speaking of getting to work, pig blood. It seems to work for some cool things, but not just pig blood, pigs. Apparently hooking damaged human lungs that have been taken out of people up to a swine's circulatory system, basically going, hey, pig, let me hook this lung up to you. It can heal the lungs, and those once damaged lungs could potentially be used for transplantation into people who need lung transplants.
Which might become very popular.
Which might become popular and very handy.
Yeah. But because I was like, but if you remove the lungs, you can't just be like, hold on, I'll give them back to you in a bit when they're better. I see what's happening here. All right.
Right. So now it's very difficult to take lungs out of a donor body and keep them healthy long enough to get them into a recipient for a transplant. We have technologies that extend the life of the lungs for up to several hours. Very often they end up damaged or they have gone past their shelf life and then they can't be used for transplants. But apparently hooking them up to pigs, the pig circulation fixes everything. Then they could be good as new. Here you go.
Can you just explain a tiny bit more what hooking them up to a pig is? I'm having trouble picturing that.
I'm literally picturing lungs outside with a little bit of tubing. couple syringes on either end, like something.
Kind of like
A
that.
brain bag almost, yeah.
The blood supply of the lungs is hooked up to the circulatory system of a pig, so that the blood then circulates through the lungs. It circulates back into the pig, gets cleaned up by the pig's liver and kidneys, and goes through the pig metabolism, and then it goes back through the lungs again. And so all of the cells and the the things that pig bodies and metabolisms do to keep the pigs healthy, it benefits the excised lungs. Pretty cool stuff.
That's awesome.
It's very awesome. I mean, if we can, anything that we can do to increase the number of organs that are available to people for transplantation is a good thing at this point in time. If you just tuned in, this is This Week in Science. Oh, hey, if you're interested in a TWIS shirt or mug or face mask, we have these things in our Zazzle store. You can head over to twis.org, click on the Zazzle link, and browse our store. Find some nice items to share your love of TWIS with the world and support TWIS at the same time. We appreciate that. Tonight, we are joined by Dr. Brian Hare. Dr. Hare is a core member of the Center of Cognitive Neuroscience, a professor in evolutionary anthropology and psychology and neuroscience at Duke University. With his wife, Vanessa Woods, he wrote The Genius of Dogs and now has written Survival of the Friendliest. Thank you for joining us tonight.
It's a pleasure to be with you, Dr. Kiki, and Justin and Blair, and your corgi, Blair. What is your corgi's name?
Sadie.
What a cutie.
Yeah.
Sadie. Hi, Sadie.
Look, she's looking right at you.
You're awesome, Sadie. I've been watching her running fast.
Okay. Brian, let me just stop you right there. Okay.
You're going to burst the puppy bubble already, Justin?
Do you feel an undue pressure having written a book about the benefits of friendliness to be friendly all the time?
My
I thought
wife.
you were going to ask about the dog book first, and then.
Okay. Wow, he just went right there. Justin, if my wife was here, she could really tell you that the Survival of the Friendliest, writing that book was a marriage. She is a very good science communicator, and I would like to think I can do some decent science, which means that if you enjoy the book, it's despite me. She can take, broccoli and turn it into ice cream, and I just mess it up and make it something that people wouldn't want to read. So we had lots of debates and battles about how to write the book. It took us 7 years to work through this marital hurdle. So she would say that I am capable of not being nice, is all I'm trying to say.
You're capable of it. but you seem like a fairly friendly guy. Is this why you like dogs so much as well?
I mean, I guess so. I do really, really like dogs. I grew up, my best friend growing up was a dog, and we went everywhere all over the neighborhood together. He went everywhere with me, and I don't know, he was just my buddy. And I was like any dog owner, wanted to know what he thought and what went through his mind and what was it like to be a dog? Was he like me or was he totally different? And so I went to college and I found out you can do this for a living. You can hang out with animals and try to figure out how they think, and people pay you for that. I am in. So that's what I've been after is, trying to imagine and learn more about what it is to be an animal and how their minds work. And then that helps us understand ourselves.
I think it does. So you've worked with dogs, you've worked with chimpanzees, you've worked with bonobos. Which one's mind do you find the most interesting?
Oh, wow. Yeah. So I've worked with a lot of different animals. I've worked with lemurs. I've worked with wolves, foxes, a whole menagerie of different primates. And I would say that I sort of gravitate to the great apes. I love dogs. They're absolutely my favorite. But I gravitate to the great apes because they just, when they look at you, you just are like, whoa. That's different. And you want to know, are you like me? I mean, are you thinking about me thinking? And so with chimpanzees, after interacting with them for about 20 years and having many chimpanzee friends, if you ask me the personality of the species, what I'd tell you is they're kind of like the cool kid in school that you wanted to be friends with, that really didn't want to hang out with you, but they were awesome. and you really wanted them to sit at your lunch table, but it wasn't going to happen. And bonobos are like the ape they're like the goofiest, silliest, like you're just like, how do you exist in terms of their personality? You're just like, I don't know, like, how did you happen? But they are absolutely, you cannot help falling in love with them. Of course, they would be the kids in school that you'd be friends with. They're kind of the goofy, like lovable, anybody can be friends with a bonobo kind of ape.
I can't quite relate to that. I went to a high school that didn't have any cool kids. There were none.
There were you were there.
No, none. We would see kids at other schools and be like, wow, they seem really cool. Nobody in our school.
You were just in a giant bonobo group. That's all.
That's nice. I would like that. I think a lot of us, I mean, we think about being in the world and how nice would it be to exist in a society of individuals that are not as aggressive as, say, chimpanzees are known for their aggression and their violence. Dogs are usually very friendly, but everybody knows, you don't trust a dog immediately. You have to get to know them. You don't know if they're going to be a nice dog or not. So how do we start looking at the traits? How are you starting to look at the traits? How did you get into digging into this?
Sure.
And the question of friendliness and the benefits of it.
Well, it's fun to be able to say what I'm going to say on a show where you guys were talking about rocket science earlier, because not all science is rocket science. And
Nope.
You know what unites science is you can powerfully attack a question and potentially falsify an idea that you think might answer it. My origin story is that I told you about my pet dog as a kid named Oreo. And I went to college and I was talking to my undergraduate advisor, who is an esteemed developmental psychologist and a very famous cognitive scientist named Mike Tomasello. And he was explaining to me that between 9 and 12 months, all of us, all humans, have a critical moment where we begin to use gestural communication and we begin to understand that others have minds like ours, and the gestures that they make are a window into their minds. So at that moment, from 9 to 12 months, that's when we begin thinking about the thoughts of others. And as we do that, that allows us to participate in culture and learn from others things that other species aren't able to learn from generations past. So it really puts us on a totally different plane, even though we have this horrifically underdeveloped brain when we're born, and still at 9 months, a terribly underdeveloped brain relative to other great apes. So he's telling me this story and I'm like, well, that's really neat. And he says, and it's completely unique that humans understand these types of gestural communication intentionally, that I'm trying to tell you what I want or what I don't want. And not knowing any better at 19, I was like, well, I think my dog does that. And that was literally the start of my career, was me saying, I think my dog does that. And so he said, well, yeah, sure, everybody's dog does calculus. And I said, no. Let me tell you the story of my dog. And I play fetch and he would lose balls and I'll gesture and tell him what direction the ball was he couldn't find. And he'd run off in that direction and orbit, look for it. And so he could tell I was serious, and he hadn't had a dog since he was a kid. So he thought, okay, well, let's go do some experiments. And at that moment, that's where I learned what science really is. It's not about being the one that has all the facts or being right. He was so excited when I came back and showed him the tapes of the dog, like my pet dog, following gestures that I was making. He went and showed colleagues that he'd been working on theories with. They're like, can you believe this? And so I remember later, after we did a whole series of experiments, we published a bunch of papers and one eventually in Science. And I remember I was interviewed by a reporter, and the reporter said, well, what do we learn from this? What have we learned from all this research? And I said, well, nothing. Everybody knew that dogs follow gestures. It was the scientists who were surprised. Apparently they don't have dogs. So but in a more serious note, after 20 years of research studying dog psychology and trying to understand how they think about the thoughts of others, we really have very carefully, and beyond my pet dog, now we've studied thousands of dogs, and we've studied all sorts of different breeds, and we've studied exotic breeds like dingoes and New Guinea singing dogs, and I've studied wolves and experimentally domesticated foxes, etc. And the summary is that if you were to ask me which organism on this planet is most like a 9 to 12-month-old child as it's first starting to participate in culture, In terms of that social skill, I would tell you it's a dog. It's the domesticated dog. It is not bonobos and chimpanzees. They can run circles around dogs on most other measures of psychology or cognition. But on that one, dogs sort of have a special ability that we think is evolved as the result of domestication.
I thought I had heard that wolves are actually quicker at reading a human gesture, getting the clue from a human looking at where the treat is and finding it and picking up on our gestures, which then made sense, like why the dog could get domesticated so easily from the wolf is because we already had somehow through similar hunting patterns or something like that. We had some sort of shared nature of visual communication between our own groups that then translated.
So in 1999, that's exactly what I thought, Justin. And we actually proposed that might be the case, that the reason dogs were doing this and great apes were not is because, why would a distant relative millions of years separated from humans have the skill that is critical to becoming human? And the proposal was, well, it must be something about wolves. They hunt and they have to pay attention to their quarry and read the social gestures and they have to understand each other. Maybe that's what it was. We've gone and tested a lot of wolves, and I've just completed a 5-year project working in Minnesota, looking at dozens of wolves and comparing. This is one of the sad stories about the research career. We were forced to work with lots of puppies, and we tested dog puppies too. And this is where I'm with Justin because it sounds really fun, dog puppies, but they're difficult. Mm-hmm. They don't sit, they don't stay. I mean, they're super cute and they try to get away with everything. You're like, guys, we're doing science. So they don't care. They don't care. And it did take us a while to figure out methodologically because our methods are very complicated. But it involves hiding something in one of two places. I don't know if you followed that. It's you hide something in one of two places and then you gesture or look where it is. And the question is, can they listen to you and realize that you're trying to communicate? And the problem when you test puppies is that you have to get down so they can see you because they're small. Even the biggest puppies that we work with, because these are 8 to 9 week old puppies I'm talking about. Super cute. Just coming up, right? Gross. So cute. Anyway, so there they are. And you're testing them. You're trying to line them up and they got to make this choice. Be serious, puppy. And what do they do? And this is when I was doing my PhD, started working with puppies and, trying to take myself seriously. And first time I gesture, I'm like, all right, what's he going to do? This is going to be so cool. Can they do it at this young? I'm so desperate to know. Or is it something that takes a lot of experience? The dog just runs right at my face and licks me everywhere.
Yeah.
And I'm like, perfect. This is going to be a nightmare. So anyway, we progressed. We figured out how to get puppies to be more serious about science. In fact, just yesterday, we published a paper on almost 200 puppies and on a whole series of cognitive tests that are service dogs. And one of the things that we've been able to use all our learnings from is to figure out how to better predict which dogs might make it through assistance dog training. And the main group we work with is in Santa Rosa, Canine Companions for Independence. So your neighbor.
And these puppies, they're smart, they're friendly. Did you find doing the work between the dogs and the wolves that there was a huge difference at the stage?
Thank you. Yeah. So I got so busy telling about how I was just trying to please Justin because I could see the skepticism and I'm like, you don't even know how right you are. But anyway, yes, what we found is that young puppies actually perform as well as adult dogs in reading human gestures at 8 weeks. And with almost no exposure to people, they can still be with their litter. They haven't gone home. They have barely interacted with people. And we compared them to wolves that we've literally been with for weeks and pull them off mom at 5 days old and have been with them for 2 months. And we play the same games. And on the social skill test, the wolves really struggle. They're great on all the non-social stuff. So if they have to remember something or they have to have some self-control, the wolves look like dogs. But on the social interaction with humans, and it's really specific to us, they're not that interested in us and they don't listen to what we're trying to communicate. So it does seem like there really is an interesting thing that's happened as a result of domestication. When we found that wolves and dogs were different and that wolves were dogs were better at reading human gestures. We kind of were stumped. We were like, okay, well, it's probably domestication. It doesn't need a lot of exposure to humans. Wolves, it's not probably inherited from wolves because wolves are not showing much skill. They look more like chimps and bonobos. But how are we going to test that it actually was domestication? So then I had this opportunity to go to Siberia, where Dmitry Belayev in the 1959 started an experiment where they selected a population of foxes to be friendly towards humans. So they kept a control line that they bred randomly in how they respond to humans. But the experimental line at 7 months, if you approached and were interested in a human, and you were friendly and wanted to basically be pet and jump in their lap, they would then breed you together with another fox who was equally as friendly. And so after just 10 to 20 generations, what they were able to show is that relative to that control population that they kept, there were increases in friendly behavior and also puppy-like behavior in the adult foxes. So they maintained sort of this whiny, puppy, cute, begging kind of vocalization. They wag their tails at a much higher rate as adults instead of the control line that goes away. And then the big shocker and the really exciting thing was they only selected based on friendliness and behavior, but the foxes also that were selected for friendliness started showing high frequency of floppy ears, curly tails, changes in their coat color, like the white spot on the forehead of many domesticated animals. And their facial features really changed. They got shorter, more feminized faces and reduced canines. So all of this happened as an accident of the selection for friendliness. I went to Siberia. I was actually the first non-Russian to get to collect data and publish a paper with the foxes. My advisor, Richard Wrangham, and this is where I got to be wrong, basically said, you have to go test the foxes. And I was like, come on, they haven't selected for cognition. Like, obviously the thing I'm interested in, natural selection selects for, like, directly. You're not going to get more cognition if you don't select for it directly, because I study that. That's what I study. I wouldn't study something natural selection didn't think was important. So he was like, it could be another byproduct. I'm like, there's no way. So anyway, I go to Siberia and he's totally right. And I'm totally wrong. It ends up that the experimental foxes are like dog puppies. They use human gestures very flexibly and spontaneously, the same way that dog puppies do. So it allowed us to see that selecting for friendliness that creates this domestication-like syndrome brings along with it an increase in social savviness.
Social savviness for each other as well. I mean, were the foxes better at relating to each other, domesticated, than they were their wild brethren?
No, it's really specific to humans. Okay. Very, very much specific to humans. The control population understand each other. They can read each other's social cues. The experimental line can do that too. There's no evidence that they differ in that way. It really is specific to humans, where fear of humans was replaced with interest and an attraction. And so they apply an old cognitive skill to a new social partner. And they can now solve new problems they couldn't solve before. It happens their new social partner is pretty good at solving a lot of problems they probably can't. So pretty good partnership.
The cognitive flexibility, I think, that's the really interesting perspective, because is it now that it's the more generalizable ability to take the skill set that they had, like you said, now be able to apply it to humans as opposed to just foxes? And does this then mean that their thinking, their cognitive abilities then become broader because they're more flexible?
Mm-hmm. Yeah. So that's exactly the idea, is that they sort of were released from a constraint of fear that was preventing them from solving or having a certain type of social interaction. They didn't have the tolerance. They didn't have the interest or the motivation to interact with humans. Couldn't solve problems in the wild, it's adaptive to just run from humans. The only problem you need to solve is get away. Because of this artificial selection, you take away that fear, and underneath it is, if you could replace that with attraction, all of a sudden you have this new ability to solve a whole set of problems you couldn't solve before. And we think that's a pattern that has repeated again and again in nature. And we've used that fox model and looked at all sorts of different species, including ourselves.
Right. And that is the key here, the idea of this self-domestication of humans, that we at some point, I mean, is this the way that your work and your evidence is pointing, that at some point we started choosing the friendlier partners. We started choosing the friendlier leaders. We started making friendlier tribes. And that what made us smarter and better at dominating the world.
In the book, we argue, Vanessa and I argue that we're the friendliest species of human to ever evolve. And in that statement, if I unpack it a little bit, there's an important new kind of revolutionary understanding of human evolution, which is it wasn't really until the last 50,000 years we were alone. There were many other humans that have big brains, were cultural, and probably were linguistic. Normally, when you'd ask a group of people in the public, oh, why are humans different from other animals? Why are we able to do it? They'd say, oh, we have big brains, we have language, we have culture. Well, that doesn't explain why we're the last human standing. Why are all the others, if it was just big brains and language and culture, we should be extinct too, or they should still be here.
No. We killed them all. Of course, because we're so aggressive.
Okay. Okay. Vanessa would like that.
I would say they're still here. Like a lot of them are still here in our DNA. I mean,
I know, I'm playing devil's advocate, Justin. Come on here.
I know you are. I'm saying the other way. I say we loved them into submission. Whoever the proto-human was commingled everyone into joining the team.
I mean, it's hard to argue with those facts. But let me tell you the angle we take on this, which is that we think that the reason we're the key to our survival and why we thrive and the other species went extinct is because we evolved to be the friendliest human, and we have a new social category that evolved that other species do not have. So our two closest relatives, bonobos and chimps, that I've spent a lot of time studying, they understand and can recognize those that are familiar as their group members. And those that are unfamiliar to them, they know are not in their group. Now, bonobos have a positive response to strangers. Chimpanzees have a very negative response to strangers. But both species use familiarity and physical appearance, facial features to recognize who they know and don't. Humans do that too, but we do something in addition that changes the whole calculus of our of our societies, or allows for society, which is we have group identity. So we have a sort of a way to recognize that complete strangers who we've never seen or met before are members of our group. And whether it's an accent or a ritual or something that somebody wears or a tattoo or even a type of tool you make, instantly you realize by seeing that marker that you actually are seeing a stranger that's in your group. And no other species can do that. And as soon as you can recognize strangers that are your group members, you can network minds together, innovate much faster, share innovations across wider spaces. And not unlike the internet revolutionizing innovation, it would have had the same impact. So technology takes off and we can outcompete the other humans potentially. Now, I know that this seems Pollyannish. Because of you already saying we beat them because we killed them, and who knows how those genes got in our species from the other species. The argument, and partly because we were coming from the self-domestication hypothesis and thinking about the cognitive architecture that humans have and the neurobiological architecture that humans have that allow us to think about others' thinking, that we sort of already covered, originates from 9 to 12 months in those gestural communication. That neurobiology and psychology allows us to have compassion and empathy for our group mates and love those that are not our family as if they are. But that same system that allows for our unbelievable and unique friendliness, it can shut down. It can turn off, and we can not apply it to all humans that we meet, and especially human groups that threaten our group or our group identity. And so that is, I think, the key of how you have this paradox solved, where we are the kindest and the cruelest species. And it's because the pressure was to be friendly, to cooperate, innovate, and have better technology. But our social identities then set up the possibility to morally exclude those that threaten our group that we love so much.
So how do we fix that? I mean, that is at the crux, right? Yeah, exactly. Magic. It's at the crux of so many issues in modern society right now, where our group identity is being used against us by propaganda, and it is affecting the way that families are able to interact now. And from your perspective, from the book, from the work, this has allowed us to survive for so long. It's deeply ingrained. How do we short circuit what we've done in modern society to be able to continue to survive?
Mm-hmm. So just quickly, the evidence for selection for friendliness, for those of you who are reasonably skeptical of this idea, the interesting thing about domestication is because it changes morphology, we could look for fossil signatures of friendliness in the faces of extinct humans. And what we saw when we did that is that human faces over the last 100,000 years have basically become, in shorthand, more dog-like. We retain juvenile characteristics, facial features, and other signals that suggest that the same neural hormones that are being affected in the same developmental pathways that are affected in dogs or the experimental foxes, or I would argue bonobos, that same process is happening in us. So then if this is true, that our nature is a friendly one, but that same mechanism that allows for friendliness, when it dampens, when it shuts off, and our empathy is like a spotlight that becomes more focused on the group we love, well, what do we do? How can we have a friendlier future? And I would say in the book, we talk about 4 options. We reject 2 out of hand. One is let's breed people to be friendly. Almost.
Yes.
Yeah.
No.
Almost. Oh, yeah. Almost. And I mentioned that because normally people who are science-minded kind of giggle uncomfortably with that. But every talk I give on this, I get a very sincere question from an audience saying, can we just breed people to be friendlier? And of course, we dealt with that in the book, The Ugly History of Controlling Human Breeding. And we then also try to, but not just do a history, but talk about the science of why it wouldn't work. It actually wouldn't even work anyway. Not to mention it's morally repugnant.
If you're breeding for friendliness in humans, that kind of, besides all of the moral stuff, there's an opportunity that you're also breeding for submission or complacency. And that could be a problem to our general society, depending on who's in charge.
And so we're not foxes. Our behavior is much more complicated. So we rule that out. And then we rule out technology. And we say technology is part of the solution, but it's part of the problem. Because the idea is the first projectile, weapon that we use, we could now hunt, and we became the atlatl or the bow. We become these super predators. And, watch out, saber-toothed cats, whatever your big teeth. Like, I got this thing. Boom. But the problem is now you can shoot and kill somebody from far away. So the same technology can be used for good or for evil, potentially. So technology is great when people are using it for great things. So I think social problems require social solutions, and that's where Vanessa agrees with me. I think I'm safe to speak for. So we really settle on 2 things. In the book, we talk about the reverse order, but first, because faster and easier, is cross-group friendships. So there's wonderful evidence that cross-group friendships, when you have people with different identities, when they are friends, it buffers, or I should say it almost immunizes the mind against dehumanization. So when your theory of mind turns off and you're no longer seeing another human as fully human, that's dehumanization. You're no longer you're no longer able to use your human unique cognition in interactions with that person. And that includes your empathy and compassion.
You suddenly become a chimpanzee.
Yeah, the worst kind of chimpanzee. And so in the extreme form, you're viewing a group of people using the same cognition you would use for a table or a chair. So what we've seen is that when you have these cross-group friendships, it really reduces the possibility of people dehumanizing each other like that. And even if one person in different groups are friends, it really affects the whole group in the network of friends that those 2 cross-group friends have. So that's number 1 is how do we have more friends across different groups, whatever those are, because there definitely is a biology and neurobiology of dehumanization, but we construct our groups and our group identity, and it's super plastic, and that's one of the things that makes it very adaptive. The second thing is democracy. And people are always sort of like, man, really? Come on. Yeah, I'm serious. And the reason I'm serious is because we didn't evolve to be despots. Our species lived as hunter-gatherers between 300,000 and 10,000 years ago, completely as hunter-gatherers who were more or less egalitarian. There was never, if you were one of my students, I get really upset when people talk about hunter-gatherers living in tribes, because tribes are defined as a group of people with a chief, meaning somebody who's monopolizing power and resources. Hunter-gatherers don't have chiefs. Democracy is the modern version of, now you've got tens of millions of people. How do we return to a more egalitarian social structure? given we have to have hierarchy and there's these different group identities, how can we make sure that when done right, the group out of power is never completely out of power, and that we're always humanizing our rivals, because even though, we may disagree, they're still important for our functioning society.
Bonobos, we were talking before the show, and you have a chapter in your book about the uncanny valley. In studying our closest primate relatives, what has really struck you about them and their cognitive abilities and their abilities to relate?
And I think what's remarkable is that they have a brain the third of the size of ours on this show, but they can do something that our species isn't capable of, which is there's no such thing as bonobocide. No bonobo has ever been observed to kill another bonobo. And so chimpanzees that are almost genetically identical to bonobos, they have lethal aggression at the rate of some human populations. They will raid neighboring territories. They will systematically, over a decade, murder all the males and then take the females and commit infanticide and basically, annex their neighbor's territory. And within the group, there's an alpha male. The first thing you do, if you're a juvenile male and you want to work your way up the hierarchy, is you start beating up your mother, because it's a safe, soft target that you can practice abusing and dominating. An alpha male, when you become alpha and everyone needs to submit to you, you must indicate your submission by kissing his testicles. This is a real thing. I'm not joking. So you have to grunt.
Dear God.
Yeah, you grunt like this, and then you have to kiss his testicles. And if not, then you'll have his fury to deal with and his coalition partners potentially. Male aggression towards females is highest when they're ovulating. Unfortunately, female chimpanzees have an honest indicator of ovulation. So imagine that there's a Post-it note when you're ovulating and you have to walk around with it. Everybody knows, today that person's ovulating. So chimpanzees, their swellings literally say within this 5-hour window, that female is definitely ovulating. So she gets targeted with horrific amounts of aggression, because the males are like, well, I'd rather beat her up than beat you up, because she's only 2/3 the size of you. And if I can intimidate her to come with me, then I don't have to deal with you. So they absolutely get coercively punished, and it's pretty rough. And so this is the darker side of chimpanzees. Now, they are also wonderful. There's also the Jane Goodall, Curious George version of chimpanzees that normally is celebrated. But the amazing thing is that bonobos that share 99.9% of their genome have none of the behaviors I just described to you. None of them.
Yeah, how do the male bonobos who run that society differ from their counterparts in the chimp side?
That's a leading question.
Yeah, thank you. I try every day to be a better bonobo male. Let's put it that way. What we think happened is there was really strong selection against male aggression, because it ends up that being an alpha is very costly. It's energetically, it's risky. It taxes the immune system, and there's beautiful data showing that alpha males really struggle to keep their tenure and to stay healthy, and there's a crash after they sort of lose that spot. So male bonobos, we think, have been selected to be much friendlier. And I'll go. I want to tell you one more fact that's fascinating before I tell you how they're friendlier. Besides saying none of that happens in bonobos, this is totally remarkable. It blew my mind. It blew our whole community's mind. It ends up that the most successful male alpha chimpanzee is not as successful at reproducing as the most successful bonobo male. And so basically, friendliness wins. You have more babies and you have more reproductive success as a bonobo male. And why are bonobo males friendly? Bonobo males, the last thing you would do is attack your mother. They are total mama's boys. They do not raid neighboring territories. They don't commit infanticide. They don't even form coalitions with each other. Never has there been a case of infanticide. And instead, if there's a problem in the group, females tend to form an alliance against any individual. It tends to be juvenile males.
Wait, it's a matriarchal society?
Okay, so
you can't.
It's not. It's not, Justin. It's not. It gets more interesting. It gets more interesting. So that is what has been a huge argument in literature. Oh, bonobos are matriarchal because if they don't have,
that's what I've always heard.
Someone's got to be in charge, right?
Okay, So what people say is, if there's not an alpha male, it must be the females in charge, says the, historically, I don't know, patriarchal society where there has to be someone in charge. So it ends up, we realized, wait a second, after watching a lot of interactions in bonobos at Lola Ya Bonobo, this sanctuary in Congo where we work, we realized, oh my gosh, nobody's looked at the babies and their dominance rankings in the dominance hierarchies. And when we did, what we realized is bonobos are baby dominant. They are not female dominant. They are baby dominant.
Babies are running the show.
Babies rule.
Yeah,
New rule,
they're baby
babies in charge.
dominant.
Wow.
And I'll tell you the anecdote of how we figured it out is it wasn't just me, it was students and this kind of thing, and talking to people who've witnessed it, but I had it firsthand happen to me. I was throwing some food into the bonobos because this is a sanctuary for orphans. It's actually, they get captive care for their life. Unfortunately, they're victims of the bushmeat trade, and we try to help the organization that helps the bonobos. And so anyway, there I am. I'm throwing food into these guys, and the food that I threw, it thuds on the ground between a little baby bonobo that probably weighed 7 pounds and a fully grown 80-pound, 90-pound adult male bonobo. And as soon as the food hits the ground, he screams at the top of his lungs and runs away. And the baby bonobo walks over and picks up the food and eats it. And so why did the male just get the heck out of there? And the reason is because I personally had this where I accidentally scared a baby bonobo, and the entire group comes to attack you.
Oh.
The entire group. And so in chimpanzees, what happens is if a chimpanzee baby is threatened, it screams, same reaction, but the mom comes, and that's it.
Yeah.
Just mom.
Mm-hmm.
And in bonobos, everybody's coming.
Wow.
And so you don't mess with a baby bonobo.
Nope.
They're just sitting there going like, we get whatever we want. And we don't know why. But and you're like, you guys are so spoiled. They're like, I'll scream. They're like, okay. Take what you
Don't
want.
scream, just don't
Yeah,
scream.
no, that would be extremely intimidating. That this little baby, they could just any second
call
have you
my
taken
family.
down by the entire troop.
Yeah. So
That
it's
is
like,
wild.
so they're baby dominant. So they don't fit. And the reason I say they're not matriarchal, I mean, I guess you could argue they're matriarchal because obviously the females are the guardians. And if matriarchal means that, females are sort of keeping the males in check. Okay, fine. But the sort of sciencey way that people think about this is male dominance typically is associated with males being larger physically. And then there are many species that are female dominant. So the lemurs at the Duke Lemur Center I work with, there are many species of lemurs that are female dominant. Hyenas are female dominant, but the females are larger than the males. And in hyenas, it's very extreme because female hyenas have male masculinized genitalia. And in fact, the same process happens with lemurs, where as females become dominant, larger, their genitalia become masculized. My colleague here at Duke, Christine Dre, is the one who has been able to document this. So anyway, along is that's not what happens in bonobos. So female bonobos are still smaller than male bonobos. They do not fit in any typical definition female dominance. And so that's why we were like, there's something else going on here. And then having interacted with them and being terrified of the babies, we were like, hey, wait a second. I think the babies are in charge.
Amazing. We know that a lot of these social differences come from cognitive differences. Cognitive differences come from neural differences.
Mm-hmm.
So, but what are some of the differences in the brain, or can you see differences in the brain between chimpanzees and bonobos? I mean, human brains are bigger, but other than that, what kind of difference is there?
Yes. So let me go back to wolf-dog comparisons and the fox comparison and the process of domestication and what does selection for friendliness do to the brain. So what we have observed, and we as the royal we, like a lot of people way smarter than me, but I get to tell you about it. What folks have observed is that when you select for friendliness, it alters first and foremost the serotonergic system. You get a much more serotonergic availability, whether that's the receptors of serotonin that allow serotonin to impact your neurons, or it's just there's more of it in
More.
parts of the brain that are involved in response to threat or fight or flight. And so it ends up that when you have high levels of serotonin, for instance, in cats, they are not particularly hungry, nor are they predatory. When that level goes down and you're in I don't know if you're Daniel with the lions, bummer. You're in trouble because their predatory behavior will be expressed. So we know that serotonin is tightly linked with aggression and predation, predatory behavior. So when looking at the foxes, they have 5 times the level of serotonin, the experimental foxes, as the control line. These are very happy. It's like being on SSRIs or ecstasy or something. And so they're pretty happy foxes. And so then when looking at bonobo versus chimp brains, some really clever people like Chet Sherwood was able to, at George Washington University, was able to show that the serotonergic axon density in, y'all probably on this show talk about the amygdala sometimes. It's an area of the brain that responds to frightening things or discrepant events. It's sort of the motivating engine of what might turn into aggression. And what you'd predict is that serotonin would have a bigger impact on the amygdala of the bonobo to sort of reduce their response to any kind of threat. And that's what we find some nice evidence for. So it seems to be the case that some of the things you would predict, and that's just one example, some of the things you would predict would be different are there. And those were a priori predictions. We didn't find it and say, oh, look, it really, people really went in.
It's not just cherry picking it out of there, right?
Well, I'm not saying we never do that, but there's definitely, that's part of science where you post hoc realize, but these types of things were a priority.
The serotonergic system in bonobos seems to be a bit more active, significantly more active than, say, in chimpanzees. In humans, we know we have an active serotonergic system. Yeah. But
so
we also have our prefrontal cortex and self-control.
Yeah, nice. Yes. Oh, wow. Thank you. That's like a fastball over the plate there. The human well, okay, great, whatever. Animals have different levels of serotonin related to their aggression. What is that? You just told me we can't do eugenics. So what does that do with humans? The comparison, it's always important. Compared to what? And so it ends up that what we think is the really relevant comparison is compared to our closest extinct human relative, Neanderthals. And what we know is that when you have high levels of fetal serotonin, it changes your facial and skull morphology. And what is the signature of our species in the fossil record? It is having a globular head. You're welcome. You all have beautifully globular heads. It is having a globular head that shows up around 300,000 years ago. And how do you end up going from having a more football-shaped or bread loaf-shaped skull like Homo erectus or Neanderthal has to this beautiful spherical globular skull that we get to carry around? You do that by early exposure to serotonin. And we know that because mice and rodents that are given early exposure to serotonin, their skulls become more globular. And SSRIs people were worried about the impact of SSRIs that were being or are
given
prescribed
to pregnant women, yes.
Who might be depressed or whatever. Mm-hmm. And there is a small risk. I'm not a doctor, so ignore everything I'm saying in terms of what you should do for your own behavior. But there is a small risk that your child, if you're on SSRIs, will have a hyperglobularized head.
A perfectly spherical head? Yes.
Yes. So we know that if you wanted to end up going from Neanderthal heads to Homo sapien heads, one pathway to do that is early exposure to serotonin. You get your globular head. That's one way we think serotonin. So the fastball across the plate, though, that you dealt me was our big brains, because Mike Tomasello, you remember the early story of Oreo and the dog, and he's excited about the experiments. Okay, so I'm sitting here, and Richard Wrangham, who really helped everything I've told you about domestication, it was together with him, my PhD advisor, and we're sitting there thinking, like, how are we? Could this really have affected human evolution? It took us 10 years to figure out how to think about it, because Mike was like, that's really great about emotions, and that you're selecting against aggression and you're reducing emotional reactivity, and you have a change in serotonergic system fine. But if bonobos are self-domesticated, like they don't have art or drive cars or anything, like I don't think it's going to have anything to do with us. And so we were really puzzled, literally for a decade. We were like, I don't know, we don't know what to do about that. I don't know. And so finally, it was the new findings about the fact, and we've already visited that we're not the only human until the last 50,000 years. And we realized that late in human evolution, there was something that needed to be explained. But back to your question about self-control. So we have this huge brain, and there's really nice evidence to show that brain size is tightly correlated with self-control. And we were able to show this looking at 36 different species, that absolute brain size, or the size of your head, is related to how much self-control you have. And it ends up that bonobos, chimpanzees, they don't have the size brain we have. They don't have the same self-control we have. So what I think happened was, late in human evolution, you already had a cultural being, a linguistic being, a being with our level of self-control, but then it was self-domesticated. And so that not only do we have massive human levels of self-control, where it's sort of like we already had this big break, but the engine got really small. We don't have the same level of emotional reactivity where we're going to impulsively be aggressive. We're going to have the same level of impulsive aggression or hostility that you would see in other species with less self-control. So it's sort of like we got supercharged by pairing amazing self-control with self-domestication.
And here we are. Let's see if we can have that self-control moving forward. Come on, people, we can do this.
You mean like wearing a mask and not going and hanging out at a pool party? Is that what you're talking about? Self-control?
Yeah, we can make our ancestors
But on the other
proud.
hand, it's been a hot summer, Blair.
And on that note, Dr. Brian Hare, thank you so much for joining us tonight to talk about your book, The Survival of the Friendliest, not just the fittest, the friendliest. Where can people find you online to find out more information or find out about your book?
BrianHare.net. The book is for sale anywhere books are for sale. And I'm on Twitter. I don't know if people use Twitter, but I do. I don't know.
I do.
Yeah, I know y'all do. I'm at BHare Dog Guy. So I don't know how memorable that is, but there you have it. So Brian Hare,
because some of our audience is just listening. Hare is Hare, because if you're thinking like hair dog or hair of the dog, it's
like a rabbit
Brian
hair
dog.
Yeah, it's like
hair,
little bunny foo. Yeah.
And we will have these links on our website.
Yeah. Oh, perfect. Just go there.
Yes.
Right there.
So if you can't remember wherever you are right now, go to our website and you'll be able to find them.
Wait, what's our website? We never even mentioned the website. That's right.
There we go. TWIS.org.
Thank you all so much.
Thank you so much for joining us. This has been a wonderful conversation.
Super fun.
Really enjoyable. But we are going to actually continue with the show because we have some other science stories to talk about. Thank you for listening to This Week in Science. You are the reason that we do what we do, that we're able to do what we do. And we appreciate that you are listening, that you're watching, that you're a part of our TWIS family. Really, it's wonderful. We love bringing you up to date every week on the latest science news and trying to give you a more grounded view of what's going on in the world of science and how it connects to our politics and who we are as people, as opposed to dredging up divisions. Let's follow Brian Hare and be friendly and find ways to humanize and have that great perspective moving forward, shall we? Head over to twis.org right now, click on the Patreon link, and choose your level of support. You can be a part of bringing more sanity and science to more people with TWIS. We thank you for your support. We really can't do it without you.
We can explain things you've heard more than.
All right, Blair, you ready for the COVID update?
boop, 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 boop. Everything's terrible.
Yeah, pretty much. No, yes. The U.S. numbers keep climbing. Deaths are going up. However, the World Health Organization suggests the public needs to be engaged in public health through individual behaviors that help others, and that the government should provide strong leadership and consistent messaging. I'm feeling a little personally attacked. Well, I guess like our country's being attacked with that message, but
Well, but we already attacked the World Health Organization.
tit for tat there, I guess.
Yeah.
Exactly. Touche. And now, good news is there are now 3 vaccines entering phase 3 clinical trials. 3 different
What does that mean?
kinds
What's
of
phase 3 mean?
vaccines. Phase 3 means it is the trial to test a very large group of people and see if the vaccine really works.
So that's the last phase before we're going to market?
Yes.
Wow.
So we have 3 vaccines. One is dead virus.
Right.
One is a viral vector, so it uses a virus that our body has that attacks cells, and uses that as a way to get into cells to deliver its vaccine message to the immune system. And then the last one is an mRNA vaccine, which is the new one. There are a couple of mRNA vaccines under development. Never been used in humans before, so we don't know exactly how that's going to turn out. But they just published in New England Journal of Medicine their report of the results, very positive, from the phase 1 clinical trials that they completed. So all of their report is finally out, and people are talking about that currently. But we'll see. Phase 3 coming up. They're hoping to get 33,000 people to sign up to take this new mRNA vaccine.
Well, thank you to all the people working tirelessly to get this thing through. And thank you to all the people who are volunteering to take that vaccine.
Yeah.
Wow.
Yeah. And if it works, so dead virus, that's pretty straightforward. That's like, okay, here's a bit of the virus, body responds. Are you going to get enough of a vaccine response? The second one, which is the viral vector, if your body has experienced that virus before, that viral vector is not going to get into your system because your immune system's going to be like, meh. So that kind of vaccine, there are pros and cons to whether or not it can actually work. The mRNA, it hasn't really been done theoretically, and in animal studies, yeah, it should work great. And their phase 1 trials looks like it should. So I don't know. Some very interesting methods out there of looking at vaccines, and hopefully a few of them will work, and that would be amazing. We will see. Other good news on the treatment side of things, figuring out who is going to become really, really sick or will end up in the ICUs. Researchers have been trying to figure out kind of that calling card of COVID, and have reported in Science this week that they think they have figured out the 2 basic traits that do indicate whether or not somebody is going to be really ill with SARS-CoV-2. And that is an interferon response deficiency. Interferon is a molecule, a protein in the body that interferes with infections. It's part of that immune response. And additionally, exacerbated inflammation. So when somebody comes into the hospital, they can test for low interferon levels, or whether or not and if their inflammation is high, exacerbated, could be something that will indicate the treatment trajectory that they need to go down. And additionally, whether or not, if it's the interferon response deficiency, then maybe they can give them more interferon. That can indicate how they can treat as well, which is pretty cool. And then in the vein of reducing inflammation, a couple of studies came out this week that were very cool, looking at a rheumatoid arthritis drug called tocilizumab. And this drug was used fairly successfully in these trials, these studies, and they weren't actual trials, but they were studies looking at groups with and controlled for without the drug. And they had marked improvements. So about 50% of people on ventilation, I think it was 45% of people on ventilation were taken off of ventilation when they were given this drug. The other paper showed that for very severely ill patients, many of them it aided their survival, so they didn't go down the path to death. So it was a really positive result. Other interesting news published by the CDC this week. They published a report finding that the use of masks by stylists at a hair salon likely influenced the reduced transmission of SARS-CoV-2 from the infected stylists to the clients. And in the report, there were two of them, Stylist A and Stylist B. Stylist A was infected with COVID initially and worked without a mask. Worked with a mask when they were with clients, but did not use a mask in between clients because they didn't know that they were infected, and then kept working after they had their PCR test, which did come back positive after 3 days. In the meantime, Stylist A infected Stylist B during their breaks because they would go on break and talk and hang out without masks. But Stylist B was also using masks. It was something they did at the salon. They wore masks when they were with clients, and nobody at the salon that was a client was infected with the virus. So it's another indication that using masks is pretty effective.
Wear a mask, wear a mask, wear a mask so you don't kill people.
Yeah. And this study also highlights that period of time in which people are very infectious, the beginning of their infection, and in which they are pre-symptomatic or asymptomatic. Stylist A at first did not realize that they were infected and didn't take care to wear a mask when not required to, and that had an effect. So there could have been other aspects where clients face away from hairstylists, so it's not like they're facing each other and breathing on each other. So maybe that has something to do with it as well. But the mask situation is highly likely to have had a huge impact there. I wanted to answer a question this week, try and go into it pretty briefly because I think it's important, and we may try and touch on it again next week. But this letter came in from Kristen Shorma from Powell, Wyoming. And Kristen said, "Dear TWIS, Kiki, Blair, and Justin, I have two very important questions I hope you will answer. One, has anyone ever actually sent you a haiku?"
Yes.
Oh, yeah. I get dozens each week. The problem is upon follow-up, it falls apart.
Haiku.
And I say, yeah. And I go, okay, so when did you come up with this haiku? Oh, well, I was driving to work and I just kind of put it together. Work time. It's like, okay, it's clearly when I request, it's a haiku that came to you in the night. This does not qualify. and then a lot of the ones that do, that are haikus that did come to people in the night are not fit to be put on the air. So it's just it's a very specific.
Very judgmental, Justin.
No, it's just a very
judgy.
specific request, and we're still waiting for one that's airable.
All right, we did air one a while back.
We did. There was one so far.
There was one.
Mhm.
Yes. Second question. What evidence is there regarding how infectious children are with COVID-19? I teach high school biology, so I will soon be in close contact with a lot of teenagers. I recently heard a claim that children harbor less viral load than adults, which makes them less contagious, but I am dubious
Smaller
about
lungs.
this claim. The information, and there was a video linked in the email as well, which I had many issues with, as a matter of fact. Kristen is a recent fan of the podcast, but you found me, Kiki, several years ago when I started teaching high school biology. I show your fermentation video to my class every year.
Oh,
It's hard to
nice.
tell if my students find it as amusing as I do, but you crack me up every time. And P.S. I love your theme music. It never gets old. Thank you, Kristen.
Thank you.
Thank you. Okay, so this week, pediatrics, a couple of pediatricians published in a pediatrics journal, the official journal of the American Academy of Pediatrics, that kids should go to school and that kids are not as much of a transmitter of COVID-19 as adults. Now, the evidence that they report in their paper does seem to show that. However, each of the incidents where they were able to study whether or not kids were the index case for an outbreak or other people becoming infected is very situationally biased. And many times schools were closing or had been closed before the opportunity for studies to take place. So we don't really have a lot of evidence, at least here in the United States, about how kids transmit. And we do know kids can transmit the virus. It's not like they don't. They do get it and they can transmit it. Now, kids have fewer ACE2 receptors than adults. And so this is one reason we think that they don't get infected as easily, that they may not they're not as likely to become infected. And so if they have fewer receptors, there's less for the virus to grab onto, and so less ways for the virus to get in. Secondarily, we don't have a lot of data on the viral load that children have or don't have. And maybe like Justin, you mentioned very, kind of softly into your microphone, this maybe smaller lungs, maybe they don't have as much air coming out. They don't, maybe
and
there are
won't
reasons.
Project this far. There's all sorts of little.
There are all sorts of things that we can come up with.
Reasons.
Exactly. Yeah. But all that said, I will point to Oregon State, the state of Oregon that I live in right now. We went from having basically no child cases to currently having as many reported cases with children of COVID-19 as the other end of the spectrum of 80-year-olds. Mm-hmm. So wow. we have increased our percentage of cases, and this could be because of the opening of summer programs and kids be mixing more freely in the U.S. than they have previously. So we are starting to see a lot more cases of children having and transmitting COVID-19 than previously.
And there's this stochastic nature of inference and who's producing and not. It's all of these things are we just don't know. And look, and they may not be a significant driver of COVID-19. Other age groups may be more significant in terms of driving the spread, but we honestly do not have a lot of data.
So the thing that I feel like people are not talking about related to this just shows that a lot of people writing these things don't spend a lot of time around children is that, sure, they could have less virus in their system, but they could actually be way more likely to spread it because of their behavior.
Yeah.
Because they could be not as good at washing their hands or distancing. They might touch their face more. They might pull their mask down more often or just have a harder time wearing masks. They might be more likely to put things in their mouth or lick things. These are all things that are going to affect transmission that are not just based in the biology of a smaller human.
I'm going to be the adult in the room. I don't mean this room. I mean the global room, the world. I'm going to be the adult in the world for just one moment, and then I'm going to stop and probably never try it again. We know that there is a life cycle of this virus within a contagious person, within somebody who has it, and how long it can survive in the environment without us. Okay. We've been going through this now for 6-7 months. 3 weeks. Time out. Everybody is grounded. 3 weeks.
Mm-hmm.
3
That's
weeks.
what New Zealand did, right? They did a month with everybody inside.
Month, but even 3 weeks should be enough time to stop it completely.
Yeah, but in terms of actual the actual question of the viral load. Children do have a viral load, even if they're mildly symptomatic. And most often, children are mildly symptomatic. They don't have the more severe stages of the disease. But the viral load follows the basic pattern, the kinematics of other age groups, which is the largest viral load is in the beginning when kids are pre-symptomatic and only very mildly symptomatic. And that's pretty much when the peak is very early. But that also that the children, and there's a study in Seoul, Korea, kept coming out of Seoul, Korea, where they looked at fecal concentration for 12 weeks and were able to find viral RNA that was detectable at very high concentrations for over 3 weeks in only mildly symptomatic and asymptomatic children. This indicates that even kids who very often don't show symptoms or don't seem to have symptoms, if they're infected, do shed virus, and that shedding of virus can transmit the disease. So part of it has to do with how many people are mixing in your community. So what is the flow of people? How are people continuing to self-isolate? If you do have kids come into school, how is the school going to manage the situation? Yeah, but like Justin said, if we could turn things off for 3 weeks, we could get a handle on it, and then potentially we could open things back up to be able to manage having school and being able to manage when someone is sick so that we don't have to worry about these questions anymore.
They don't
I
care.
I hope that addressed your question a bit, Kristen. Speaking
can
of
I
Brian
take a
the
stab at it
dog
real
guy, how about speaking of Brian the dog guy, Blair had a dog COVID story.
Oh yeah, you know how back in the spring everyone was like, "This one dog had COVID." We think dogs can get COVID.
I remember cats. I don't remember dogs.
Yeah, it's not real.
Okay.
University of Colorado was the main home of this study, but it was actually an international collection of scientists, and they looked at the study published earlier this year claiming coronavirus may jump from dogs to humans and found it to be flawed, have no direct evidence to support its conclusions, and that basically it was all just based on the fact that a distantly related dog coronavirus has a similar CpG content, part of the coronavirus genome, as SARS-CoV-2. And because this distant virus replicated in the dog's digestive tract, the conclusion was that dogs' intestines were a great place for SARS-CoV to live. But that's true of all mammals. So not really a compelling argument.
No.
Ultimately, dogs are not a more plausible host than any other mammal based on current data, and in fact, far less than bats or pangolins. So your dog is fine. Your dog is not going to cough on you and give you COVID. Don't worry about it. Still, if you pet a stranger's dog, wash your hands before you touch your face.
If you just tuned in, you're listening to This Week in Science. Want to help TWIS out? Leave a positive review for us on your favorite podcast platform or video watching place where TWIS is today. Hey, Justin, do you have a story?
I got one. I don't even know anymore. So this is a quick one. Study in the journal Leadership and Organizational Studies by San Francisco State University. They looked at, this is using data from the Fullerton longitudinal study. They started tracking families and asking them all sorts of annoying questions back in 1979. And a fun thing, correlation that came out, that they saw that, this is quotey voice of Herbert Walker, not Bush, but Dana Herbert Walker, says, "We see it all the time where their obnoxious leader rises to the top, but we don't know much about why. Tyrants, whether they be in the boardroom or in politics, wouldn't have the power they do if followers didn't support them. We often look to leaders to explain leadership, but we should actually be looking at their followers." And what they found in this correlational study is that people who had a lot of conflict in the home as youths tended to support tyrannical leadership styles as adults. So the first of all, it might explain support for our current leader, which just says something terrible about the childhoods of those people who are supporting. But the president himself apparently had a Klansman and apparently a pretty domineering father, or Klansman for a father who was apparently pretty domineering over him as a child. And our own president is constantly talking about his admiration for dictators globally. Maybe that explains it. Maybe that's just it. It's bad parenting that leads to tyrants in society. Yeah, I just thought it was funny. But I thought it was funny that they're looking at the followers and who had conflicted with the parents and had overpowering, dominating parents, and then those people sort of people who are being more tyrannical in places of power. But it immediately made me think of our own president's admiration for all. Like, why is the guy who made his coin and was, or whatever, in a free society with capitalism and everything else this guy going, why does that person admire all of these dictators? Oh, yeah, it may be nothing.
Justin, I don't think he actually made any.
He didn't.
No,
he lost all the money.
He lost
So I feel like this is
spent
a
a lot of
problem.
money.
It's fine. Don't worry about it. It's all moot.
Did you have another story, Justin?
A compound that is commonly found in pickled capers activates proteins required for human brain and heart activity.
Wait, what?
In a pretty direct way, yeah. It may even
I feel
be
like you just said a bunch of words that didn't fit.
I did.
It went from pickles to brain and hearts.
Okay, I'll slow it down. I know it's late.
How is this direct link? Tell me. Explain.
A compound commonly found in pickled capers has been shown to activate proteins required for normal human brain activity and heart activity, and may even lead to future therapies for the treatment of epilepsy and abnormal heart rhythm. This is the universities from University of California, Irvine, discovered a compound named quercetin, commonly consumed when you eat capers, can directly regulate proteins required for bodily processes such as heartbeat, thought, muscular contraction, and functioning of the thyroid, pancreas, and your gut. So, but that's what's interesting about this is just from the cursory level of like, hey, we found a food that has a precursor that then gets broken down and built up and turned into something else that gets used in the body for is normally what you're hearing from a nutrition story. Like if you eat this is a building block that an enzyme, if it encounters, is going to turn into a thing that does a thing that does a thing. This is saying there's a direct action, direct stimulation to these proteins, which is pretty unique in a nutrient derived from food story. The Abbott Lab, where this was discovered, found that quercetin, plant-derived bioflavonoid, modulates potassium ion channels and the KCNQ gene family. These channels are highly influential in human health, and their dysfunction is linked to several common human diseases, including diabetes, cardiac arrhythmia, and epilepsy. Study revealed that quercetin modulates the channels by directly regulating how they sense electrical activity in the cell, suggesting a previously unexpected mechanism for the therapeutic properties of pickled capers.
That's amazing. And I'm looking further in this story, and apparently we've been eating capers for about 10,000 years.
Well, I haven't. They're gross.
And humans have been using capers in traditional folk medicine for thousands of years.
Wow.
Capers have a history, which I had no idea about.
You know what?
I've never asked for a caper, pickled, or otherwise on my pizza.
Pizza.
Or like have a caper. I don't know, where do you put a caper?
I mean, I've
don't
only
even
ever
know.
seen them
Pasta.
on
I
lox
put-
and bagel is what I've seen it on.
Logs and bagels. I make a nice
there.
pasta with green olives, a tomato sauce with green olives, and capers. Very
I'm
good.
putting them on all my pizzas from now on.
Apparently, however,
Oh.
quercetin is not only in pickled capers, also in abundance in apples,
Oh.
honey, raspberries, onions, red grapes, cherries, citrus
Oh, I like
fruits,
those.
and
I like
green
those.
leafy vegetables.
Oh, great. Who needs a caper? Get out of here, capers. Nobody asked for
So-
you.
Quercetin is found.
Gross peas or something. I don't even. What even
Gross.
are you? I don't know. Justin, did you realize you were hitting a nerve with this one?
I
Because
had no idea.
No clue.
I'm offended by their presence when they show up on something.
The number one most quercetin dense vegetable is called dock. Watercress, cilantro, radicchio, asparagus, okra, onion, serrano peppers, red leaf lettuce, kale, elderberry, cranberry, blueberry, blackberry, fig apple.
It's just in all plants?
It's a plant.
Yeah,
Yeah,
all
it's
plants.
it's in plants.
It's in
all the
food.
plants.
Don't worry
You
about it.
just
It's in
got-
food.
All right, moving on from pickled capers. That was a pickled caper. Or was it just a pickled caper?
Ooh.
Oh. Sometimes we just don't see things the same. We don't see eye to eye. And, you notice that anyway. Researchers really found that. Publishing out of UC Berkeley in the Proceedings of the Royal Society B Biological Sciences. Researchers had people look at a visual frame and look at arcs and the different length arcs and size of arcs, and had people compare them, and looking at the differences between different performances by different people, they found that people found the exact same arcs, that measure a very specific distance, to be bigger in some locations in their visual field and smaller in other locations in their visual field. And there was variation in where those locations were between people. So really, what it suggests is our eyes and our brain basically perceive things in the environment, and none of us are perceiving the exact same thing, but we all have to use our physical body to adapt to work with those things. And the example they give in the story is, say, a mug of coffee. The mug of coffee is the handle to that mug of coffee is always going to be the same size. It's always going to look the same size. But regardless of whether I'm looking at it out of my side eye, giving that coffee a side eye, and I'm going to go reach for it and grab it, I'm going to be able to reach for it and grab it and be able to hold on to it and get that coffee to my face hole, regardless of how, I'm going to figure that out. My body's going to figure that out. And if it's the same distance, anybody's going to be able to reach out and grab that handle of coffee, be able to get that cup of coffee. So our bodies adapt for those differences in how we perceive the things in our environment, but none of us are necessarily seeing things in the same way. We may have a visual perception fingerprint that is unique to each of us. Trippy!
Well, we know we don't see color the same.
Well, I certainly don't.
Yes, no, you do not.
But even those of us who aren't color different, how do you say it now? Not challenge. It's just color unique, color different
Yeah.
different seeing.
Yeah,
I don't know.
sure.
I'm gonna get in trouble.
Differently seeing. Yes.
Even those of us who aren't are still like with variations. Like we're a lot of times people there's studies that can show that or not even studies. There was an experiment at the Exploratorium in San Francisco. There was like this wheel chart, and you would match up, and it would suggest you do it with a friend and see where you match them up. And quite often you're like, these two are the same, and the person next to you or the person before you would match that wheel up slightly differently, like a shade differently here or there. So we perceive light differently, and that creates a difference in how we perceive shade or coloration and hue. But also, I kind of wonder, like, if there's anchoring involved in any of this. Like, when you're talking about whether it's a big arc or a small arc, if there's like, what was the last arc that you saw? Was it? Did you just visit St. Louis or McDonald's or yeah, people, we're all living in our own worlds. This is just
we're all living in our own worlds, but
giant
sharing
game of
the
telephone.
Same world. Exactly.
Big game of telephone.
Big game. And then my last story for the night is about the placenta. So your seahorse placenta story intrigued me earlier, Blair, because in this new study, researchers have determined some very, very important information about the formation of the human placenta. And in this research, which has now been published in Nature, they looked at the level of the nucleus of the cells that go on to become the placenta compared to the cells that go on to become the baby. And at the moment of fertilization, there's division, and some cells go to become the placenta and the others become the fetus. And the placental cells, within the DNA, there is a twisting that happens. So the DNA starts twisting up really hard and it breaks. And so the DNA breaks out of its double-stranded formation. You know, normally it's like a little twisted ladder, but instead it gets twisted and broken into single strands. And it is the fact that it is in these single strands that allow it to go on to become the placenta, and also potentially lie at the heart of diseases like preeclampsia, or at disorders that lead to malformations of the placenta, or that lead to overgrowth of the placenta, or just to a very nice placenta that feeds the offspring. Yeah, so the placental growth is regulated by a sixth base of DNA called N6-methyladenine. And this base stabilizes these single strands so that they stay as single strands. And there is a protein around that's called SATB1, SATB1. And SATB1 is critical for the organization of chromatin. So it takes things and puts them together and makes them become double-stranded, right? Chromatin, got to wrap it up. SATB1 wants to do that. And so when there's none of this N6-methyladenine, the sixth base of DNA, placentas grow uncontrollably. But if these, and so they have defects. And so they now want to look into the interplay between this DNA base and also this protein, and how these single strands of DNA actually function to make end up determining the fate of the placenta. And it's this pretty cool cellular level research that will hopefully lead to treatments that can help many more healthy babies be born. Oh my goodness, is it time for the animal corner?
Yes.
Did we get to the animal corner? Is it time?
Yes, it's time.
Okay, this is This Week in Science. It's time for Blair's Animal Corner.
With Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl.
except for giant pandas
And capers.
Capers aren't animals, Justin. So my first story feels like I prepped this story a month ago. It's about rats. Oh my gosh, I'm falling apart. Rats, and how they decide to help or not help a neighbor if they are in distress. It turns out that rats actually exhibit the bystander effect, which is a phenomenon in which individuals are less likely to help someone in need with other people around. This is something that was originally seen in 1964 by humans in a really sad scenario, but ultimately they showed that if there's a bunch of people around, there's kind of this moment of like, are you going to do it? Am I going to do it? Nobody's going to do it. We're not going to do it, sort of thing. So these rats were less likely to rescue a rodent in need if they were surrounded by unhelpful bystanders, but they were more likely to take action when their fellow bystanders were willing helpers. So it's, if somebody goes in there and says, you call 911, I'm going to do CPR, help me out with the compressions, then people are in it, right?
Yeah.
So it's this idea that it just really kind of feels like a herd mentality thing. But these rats definitely did it. The way that they investigated it is pretty interesting. They wanted to have this situation where there were helpful rats and unhelpful rats, and you kind of watched what this other rat in that space was doing. So they had to make unhelpful rats. So how do you do that? You don't want to anesthetize them. You don't want to incapacitate them. So instead, they gave them a low dose of anti-anxiety drugs to basically just reduce their level of interest in helping, just kind of make them kind of spacey. Yeah, exactly. So these incompetent helpers were put near a potential helper that encountered another rat that was trapped. In contrast, rats were paired then with helpers that consistently failed to release the distressed rat. And so incompetent bystander rats influence the non-impaired bystanders if they belong to a strain of non-impaired bystanders they were familiar with. So if the people who were not helping were people they knew, not going to help. They're not helping, I'm not going to help. So this kind of, it shows that it's a little bit more complicated than just, are people helping or not? I'm going to do what they're doing. It actually does, there's familiarity involved as well. But it also undermines the original thought about the bystander effect, which was that it has to do with individuals assuming that others will be responsible for taking action, which lessens their own sense of responsibility. So their thought here is because these rats are doing this in this kind of very simplistic scenario, they're not communicating with each other. So it's clearly not a situation of, are you gonna save that rat or should I save that rat? And then it just kind of turns into no action at all. Instead, it looks like this is just a more broad mammalian process, or at least that's what they're claiming in this study. So I'm not sure if that's such an easy leap here. I do think that animals pick up on each other's cues, and if nobody's helping, there is kind of a moment of like, am I not supposed to help? Maybe I shouldn't help. You're not helping. Maybe I should. So there's definitely some very funny cognitive stuff happening here. It's interesting. It happens in humans and in rats. But yeah, I'm not really sure what exactly it means based on this study.
Yeah, I mean, exactly. What we can't look at it through the lens of our human interpretation either.
Right.
Yeah.
What we know is helpful rats, test rat is helpful. Unhelpful rats, test rat is unhelpful, but especially if they know those other rats.
Well,
If
it's
you-
very similar. I'd like to talk to Dr. Brian Hare about this study.
I know. I was
and-
kind of hoping he was going to be around for this one.
Why did he ever leave?
So
He should never have
You
left.
should have, because
So,
it was
I
1 in
mean,
the morning.
to kind
That's
of
no excuse.
to piggyback on that, I think what this does tell us is if you see someone in peril, in pain, who needs help, and no one is acting, be the person to act, because if you act, then other people will jump in,
Especially if they know you.
especially if they know you. But yeah, it's this, there's a paralysis that happens if nobody's doing it.
Yeah.
And that's ingrained in a mammalian history. So just suck it up and be the first person to help.
I think that goes is pretty deep in it, is the fear to be out of the ordinary, because the ordinary is safe. The status quo is safe.
Right.
Whatever the rest of the rats are doing is what's going to help you survive. When you are the one rat in a social setting that's going to go help a rat who's in trouble, you could end up in trouble too. What did that it's fear, it's survival. And I think that's a very interesting point. And I wonder what the trut foxes would do.
Yeah. Well, and the other thing that I just realized is it could totally be a prey instinct, because if a member of your group is injured, that's the one that's going to get eaten by a predator right away. If you go to rush to aid that predator all by yourself, you will also get eaten.
Right. Mm-hmm.
Yeah,
I think that's a very good point.
Yeah.
Mhm.
And if all of you go over there and protect the injured individual, then it's much less likely you'll all get eaten. So that might have something to do with it as well. Okay. But really, that was the appetizer. I have to get to this story about left and right handedness in fish genitalia. Okay.
Okay. Wow. All right.
I've been waiting to talk about this. So
Go
this is
for
a
it.
study from the University of Constance, and it is focusing on a century-old question regarding asymmetry in genitals in internally fertilizing fishes. Which, okay, so first of all, we know most fish are external fertilizers. The females release eggs, the males release sperm on top of it. But there are some fish that internally fertilize. For example, sharks. And they have a phallus-like thing called a gonopod that's basically an anal fin that has transformed through evolutionary history to be a copulatory organ. And they use that to inseminate females. So those are the internally fertilizing fishes. So let's get that out of the way. Now, let's talk about the fact that their genitals are asymmetrical. So I'm about to throw something else at you. This is specifically looking at the genus Anableps, which is four-eyed fishes. So four-eyed fishes.
Wait, are you about to throw the asymmetrical genitalia of a four-eyed fish in my direction?
I sure am
right now.
So a four-eyed fish basically has a quote-unquote split eye. So it's really they have two eyes, but they have a section of it that can see above water and a section that can see below water. So it looks kind of like they have four eyes, like they're kind of funny shaped. They're wild animals. But anyway, they're these four-eyed fish that are really two-eyed fish. They are live bears, as I said. They have an internal fertilization. They give birth to fully developed offspring. They do not lay eggs. And they have this asymmetric genitalia. Both males and females, by the way, have asymmetrical genitalia. So these gonopods that I talked about before, they're asymmetric. Their tip either bends pretty drastically to the left or to the right. Some females have asymmetric overgrowth that covers their genital opening laterally. So some females can only copulate with some males. So within populations, you see males and females that are both righties and lefties. You would guess that over time, this would lead to speciation. Because if righty males and righty females are the only ones that are compatible, that would continue, right? And eventually you'd have your righties and you'd have your lefties, and they would be separate lineages, separate species, completely distinct. Well, in order to figure out if that was true, you would have to figure out if this is a genetically marked trait. How do you do that? You look at the genitals of mom and dad and the genitals of their babies and see if they are the same. Do left-sided males father mostly left-sided sons? So when you look, you have individuals with left or right-sided genitalias found in similar proportions within populations. So one is not dominant, even if it is genetic. But they found it's probably not. There is no strong heritability in the direction of genital asymmetry in these buddies. So it seems to be 100% luck of the draw, random. Both captive and natural populations had offspring where they were asymmetric and a near equal proportion of left and right-sided genitalia. So, and all this happened independent of their parents. So that explains why this continues, despite the fact that it kind of pigeonholes you into a specific pool of mates. But so yeah, it looks to be completely random. There is no evidence of genomic differences in them at all that they could see that could be linked to it or anything. And so therefore, they don't think there's any way that this could lead to speciation in these guys. So this is just a really bizarre situation where it appears to be just a flip of the coin. Are you going to have left-sided or right-sided genitalia? And it might narrow the dating pool, but other than that, it doesn't seem to affect their lives at all. Which, of course, then you have to ask the question, why do they have those then? Which we don't know. That's next, I guess. We have to figure out what drives that. What drives the left or right-handedness? Is it genetic, but it's just a flip of the coin? Is it hormonal? Is it just like how you sat inside the mom? We have no idea how the handedness happens. But I'm guessing that's next.
Randomly.
Mm-hmm. We don't know.
It's great. I mean, when you go in, researchers go in and think, this is definitely going to be a genetic thing. This is like a sex-determined trait or something. But, we're going to do this study. It's going to come out this way. Nope. What is going on?
Nature's just crazy.
Mm-hmm.
Nature's crazy like that. I wonder what does lead to it.
Well, maybe we'll find out. I'll have to keep
Someday.
an eye out for the four-eyed, internally fertilizing, handed genitalia having fish.
You keep an eye out for
I will.
the fish genitalia, Blair.
You will.
Mm-hmm.
You know I will.
I know
All right,
you
girls,
will.
I think we're all getting a little loopy. You've been talking about fish genitalia for, I think, 20 minutes now. What's the next story you got, Blair?
I'm done.
Oh.
We've made it to the end of the show. Great show, everyone. That was fun. Thank you. Thank you for listening. I hope that you enjoyed the show. And thank you for our guest, Dr. Brian Hare, for joining us this evening to talk about his book, The Survival of the Friendliest. It was a great conversation. And you can find out more about him and the book that he's written with his wife, Vanessa Woods. And we will have a link at our website with our show notes. So shout outs. Fada, thank you for your help with social media and show notes. Gord, thank you for manning the chat room. Identity, thank you for recording the show. And I would like to thank our Patreon sponsors and the Burroughs Welcome Fund for their generous support. Thank you to Eric Combs, Flying Out, Guillaume, John Lee, Ben Rothick, Ali Coffin, Maddie Perrin, Taylor P.S., Josiah Zahner, Sarah Forfar, Donald Mundis, Rodney Lewis, Stephen Alberon, John Ratnaswamy, Dave Friedel, Daryl Myshak, Stu Pollock, Andrew Swanson, Corinne Benton, Sky Luke, Paul Ronovich, Ben Bignell, Kevin Reardon, Noodles, Jack, Sarah Chavez, Paul, Jason Olds, Brian Carrington, Matt Bass, Joshua Fury, Sean Lamb, Sue Doster, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, Ken Hayes, Howard Tan, Christopher Rappin, Richard Hendricks, Brendan Minish, John Gridley, Richard Porter, Christopher Dre, Mark Mazzaros, Artyom, Greg Briggs, John Atwood, Robert, Rudy Garcia, Dave Wilkinson, Matt Sutter, Philip Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jim Drapeau, Alex Wilson, Dave Neighbor, Krusty Ranky, Matthew Litwin, Eric Knapp, E.O., Kevin Parachan, Andrew Louthan, Steve DeBell, Bob Calder, Marjorie, Paul Disney, Dan K, Patrick Pecoraro, Gary S, Ed Dyer, Tony Steele, Ulysses Adkins, Brian Condren, and Jason Roberts. Thank you for all of your support on Patreon. If you are interested in supporting us, you can find more information at patreon.com/thisweekinscience or click the Patreon link at twist.org. On next week's show,
We will be back 8 p.m. Pacific time on a Wednesday talking about sciencey things.
Yeah, go to twist.org/live. If you want to listen to us as a podcast, you'd want to just have us in your ear holes instead of also in your eye holes. Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get some friends to subscribe too.
Yeah, you can also hit us up on the Twitter where we are at JacksonFly, active Dr. Kiki, at what is it? At Blair's Menagerie?
Yeah. Also, if you want information on anything you've heard here today, you can see notes and links at www.twist.org. You can also sign up for a newsletter. And if you want to shoot us an email, you can email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair, me, at blairbaz@twist.org. Just be sure to put TWIS, TWIS, in the subject line, or your email will end up, I don't know, in a pile of dog poop, I guess.
Eaten by a dog.
Yeah. We'll be back here next week, and we hope you'll join us again for more great science news. And if you've learned anything from the show,
if you learned anything from the show, please remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. 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 And This Week in Science is coming your way. So everybody listen to everything we say, and if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a
a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness, I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science, This Week in Science, This Week in Science, 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 This Week in Science, This Week in Science, This Week in Science, 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 This Week in 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 Science.